Welcome, folks, to our second session. The talks in this session provide overviews of the 6 families and over 560 species in the snapper seabream grunt specialist group of the IUCN, which you've learned a lot about, hopefully, in the first session. These species can use habitats from freshwater deep reefs in primarily tropical and subtropical, but also substantial temperate regions as well. Diversity in numbers of spawning aggregations by SSG families and regions will be estimated based on variable information, much of it from Fisher's knowledge in this session. In addition, FSA environmental drivers and the roles of geomorphology will be considered. And we'll end with examples of successful management of species with spawning aggregations presented from Belize and also Western Australia. These are very, very interesting talks, and we look forward to sharing this with you and getting your input. The session will end with a 30-minute question and answer as well. In our first talk today, Dr. Melita Samoilys, Director at Cordio East Africa, will provide a biological overview of life history strategies of the species in the IUCN's snapper seabream and grunt specialist group.
Hello everyone, welcome to this talk in session 2. I'll be giving you a brief biological overview focusing on life history strategies found in the snapper and associated families of this species specialist group. I'm Melita Samoilys. I'm a co-chair of this specialist group with Ken Lindeman, and I'm also a director of Cordio East Africa based in Kenya. In this subsession, I want to give you a short introduction on the diversity of life history strategies that we find in the snapper and associated families of this IUCN specialist group. And I'll focus in on reproduction. It's one of the key life history strategies in fishes that can have huge implications in terms of their vulnerability to fishing. And this relates to the topic of this whole workshop, that of spawning aggregations and why they need conservation attention. I'll also touch on their ecology, such as their habitats and their movements among those habitats. And I'd like to remind you that many of us are working in a context of inadequate management or data-limited management in multi-species fisheries, where these SSG species are frequently captured. So linking life history strategies with vulnerability is an important approach for managing fisheries, and I'll touch on that as well. So what do we mean by life history strategies? I'm going to give you a very rapid overview on what is a really huge topic. Age, growth and reproduction are the 3 primary life history parameters that fisheries scientists consider in examining management approaches. How quickly do fish grow? How long do they live? How and when and where do they reproduce? How fecund are they, etc. And I'm sure you've heard of R versus K life history strategists, which is a shorthand for those that have a short life, rapid growth, rapid reproduction and high fecundity, such as sardines, versus long life, slow growth, slow reproductive cycles, such as sharks. Very different strategies in terms of life history. So for this workshop, reproduction is obviously a key life history strategy for us to focus on. But also it is closely linked to how resilient a species is to fishing. So this table gives you a quick summary of what we know about the reproductive strategies of the 6 SSG families. You can see that the diversity of strategies in these taxa is astounding, both between families but also within families and even within a genus. Phylogenetic linkages can explain some of this. For example, bream, both Sparidae and Nemipteridae, and emperors are closely related, and they're strongly hermaphroditic, though some of them are also gonochoristic. Snapper and grunts and fusiliers, though, are strongly gonochoristic. FSAs are found in most families. We don't know very much about nemipterids. And there's still a lack of data on a lot of these species. Species that form FSAs can be either gonochoristic or hermaphroditic, but being hermaphroditic generally makes a species more vulnerable to fishing. Finally, we list a summary on the occurrence of migration between habitats, which is a common feature of many of these families, but information is still lacking on that. Moving on now to some specific examples to explain this diversity in life history strategies a bit better. There's a huge amount of literature on this, so I've just picked out a couple of examples. And this slide on emperors, the Lithrinidae, comes from a study on Australia's Great Barrier Reef led by Curry in 2013. And they looked at various life history characteristics of 6 species of Lithrinus, and they found they group into large and smaller species based on maximum fork length. However, longevity or lifespan was not correlated with size. Growth was rapid and similar across all 6 species in the first few years of life, but then it differed, so the growth coefficient differed. In terms of spawning seasons, we can see these 6 species, across the 6 species we've got winter spawning, summer spawning, as well as all the year round. Turning to the ecology, particularly the use of habitats and movements, we've used an example here from snappers and grunts in the Western Atlantic, and there are over 45 species of these 2 families there, and they exhibit varied behaviour with respect to their habitats and movements, which is seen in many species, but not all. They can occupy shallow and or deeper habitats across the shelf. Some of them form feeding migrations from shallow seagrass, for example, and mangroves to coastal shallow reefs, and that's an important ecological function in terms of transferring nutrients. And several species exhibit ontogenetic movements between different habitats. A well-known example from the Indo-Pacific is Janus argentimaculatus, the mangrove jack, where juveniles recruit into mangrove habitats, and then as adults they slowly migrate as they get bigger further offshore to coral reef habitats. A final example of diverse use of habitats and movements for spawning comes from the Sparidae, the bream, sea bream, and this is shown here in these graphics from a recent paper comparing 8 sparids from South Africa by Whitfield and Mann, and you'll be hearing from Bruce Mann in session 4. Firstly, we can see in the top graph that the 8 species fall into 4 groups in terms of their association with estuarine habitat. And some of these differences are within genera. Secondly, even within the highly estuarine associated group, and you can look at the graphics on the bottom right, that's Acanthopagris vagus and Raptosargus holubi. There are differences in how these 2 estuarine species use estuarine habitat for recruitment and spawning. And what this illustration shows that Raptosargus holubi spawns near estuaries, while recruitment occurs in the surf zone, whereas Acanthopagris vagus spawns in estuaries and near shore, and recruitment also occurs in estuaries. Understanding these ecological and life history aspects of the species you're working on or managing is clearly vital. The final topic I want to touch on is understanding species vulnerability to fishing and what life history strategies make fish intrinsically vulnerable to fishing. I'm presenting here a vulnerability framework we used in Eastern Africa for species that form FSAs, not SSG species here, I'm afraid. And this is captured in this book on FSA research for management that Jan Robinson and I edited in 2013. It's available on the Hoover Toolkit. I also flag a recent paper using a similar approach in the Gulf of Mexico by Biggs, led by Biggs with Will Heyman and Jan Robinson. So to assess how vulnerable a species is, and therefore in turn how resilient it might be to having its spawning aggregations fished, depends on 2 sets of vulnerability indices. The intrinsic indices, which are population-specific life history characteristics, that's what we've been talking about, and then extrinsic, which is the level of fishing that the species is subjected to, as well as other threats, such as poor management, no MPAs, etc. So here are the intrinsic vulnerability indices that we used in the Eastern African research example. For example, you've got the top rapid growth means you're less vulnerable. That's typical in rabbit fishes. Maximum length, generally the larger the fish grow, the more vulnerable they are, but it's not always the case. High natural mortality means less vulnerable. And what I've circled in red just to highlight them are those that make you vulnerable to fishing, more vulnerable. Large size, long life, and sexual maturity at an older age are vulnerable attributes. In terms of hermaphroditism and gonochorism, hermaphrodites are more vulnerable. And in terms of the type of FSA, which you'll be hearing about elsewhere in other talks, transient spawning aggregations are more vulnerable. This slide shows the extrinsic indices in the table, and these relate to different aspects of exposure to fishing, and then the final vulnerability matrix in the plot on the left. And this was an assessment we did for groupers and rabbitfish FSAs in Seychelles, Kenya and Zanzibar. And in the plot on the left, you can see the vulnerability matrix of the intrinsic against extrinsic indices and where the different species we were doing our research on fall in this matrix. The blue circle is the best place to be. In summary, I hope this short talk gives you a good sense of the importance of understanding life history strategies of the species you're researching or managing, and that it will also help answer the question, can we really fish spawning aggregations sustainably? Of course, the opposite of vulnerability is resilience, and this is a crucial topic, as you all know, in the 21st century. Thank you.
Thank you so much, Melita, for that summary of such a highly diverse group of families in terms of so many different metrics. Very appreciated. Our next talk is by myself and Sarah Brooks of Florida Tech on spawning aggregations of snapper, sea bream, and grunt specialist group families. Hi, and welcome to our presentation on spawning aggregations of snappers, sea brims, grunts, and associated families with a focus on patterns among regions. This outline slide will occur 3 times, indicating 3 different components to this talk. This first component is a brief introduction, review of the systematic literature review and validation rankings that were developed by this project. SSG species includes 6 families with 561 total species. Again, SSG species are snappers, sea brims, and grunts, plus 3 other families shown on the right: the threadfin brims, the emperors, and the fusiliers. The name for the specialist group in the IUCN that this represents just couldn't handle 6 family names. Overviews of the species diversity of this immense and wonderful and important array of beasts, their importance to livelihoods, their life history patterns, have been provided in talks in session 1 and 2. Very briefly, the systematic literature review discussed here was developed and implemented and analyzed primarily by Sarah Brooks, graduate student at Florida Tech master's program who just graduated early May 2024. The literature review focused on Google Scholar and SURFA as the 2 primary databases, SURFA being Science and Conservation of Fish Aggregations. Many of us know SURFA because of their outstanding resources for many, many years, including their FSA database. So Sarah conducted a systematic literature review using tools used in metasynthetic literature studies. The systematic review flowchart is shown on the left, screening and inclusion and exclusion criteria on the right. Of note on the left, she identified an N of initially 637 records from both databases combined. That's the top left box. And then on the bottom left box, ultimately, after doing the criteria involved, processing those criteria, 71 studies were included in this review. There were so many studies that were identified both from the Google Scholar search and in the SURFA database that post facto we determined that we would develop a FSA validation rank approach wherein 3 ranks would be assigned based on the level of confidence of the FSA data. The working criteria for this are shown in the right side of this table. 1 direct indicator from those primary sources for rank 3, 2 indirect indicators for rank 2, 1 indirect indicator or other evidence for rank 1 for low, medium, and high 1, 2, 3 relative levels of confidence. What patterns were developed from this study? Or I should say identified. The total SSG spawning aggregations by spawning validation rank are presented here for Google Scholar and SURFA databases combined. And as you see on the bottom right table, the total number of FSAs identified were 326 for snapper, seabream, grunt, emperor, threadfin brim, and fusiliers, with the threadfin brims not having any identified yet. All the other families do. More on that coming. So the FSA validity ranking by SSG family from Google Scholar. These would be the new records that are, in almost all cases, I believe, are not in the SURFA database in this presentation. Luchonids have the most high confidence ranks and also the most low ranks, as you can see in the highest bars in the plot. There are many potential Lithrinid FSAs. Most are ones from Fisher knowledge sources with a lot of potential to be developed further in terms of validation. Most potential or confirmed FSAs are in a small number of genera: Luchonus, Lithrinus, Plectorhynchus. Most species form transient FSAs, but some form resident FSAs, including some important Luchonus species and some possible Hamulid species and others as well. Validation ranks regionally with both databases stratified show that the West Atlantic to the far left has the most high confidence rank FSAs. Google Scholar shows new potential FSAs in terms of regions for the Mediterranean and South Asia. In looking at this and plotting this for this presentation, we are definitely going to generate in the next version of this a plot with all the databases combined. Scaling down to an important family, the Luchonid FSA validation ranks for SURFA and Google Scholar sources, shown here by region, by database. The most potential and confirmed Luchonid FSAs are in the West Atlantic and the Northwest Pacific. This is the Coral Triangle above the equator. In addition to the West Atlantic, Google Scholar has many 1 ranks in the WIO, West Indian Ocean, and the NW, Northwest Pacific, most from Fisher knowledge, shown in the plots in gray for Google Scholar as 1s. Next, we're going to look at a regional example of Luchonid FSAs in Brazil. These 2 slides were provided by Dr. Beatrice Padovani Ferreira from the University Federal de Pernambuco, Universidade. They have conducted some remarkable work on mutton snapper and dog snapper along the northeast Brazilian coast using catch per unit effort, gonad histology, and also GSI. A lot of this is summarized in the paper on the top left. The citation is on the next slide. As an example, you can see Brazil northeast in the image to the left, and then on the right, the spawning sites of 2 areas within that northeast coast. Yellow circles represent mutton snapper, purple circles represent dog snapper. These sizes are proportional to CPU increases during spawning. They've done a lot of gonad histology, and these photomicrographs show the presence, especially on the right, the presence of post-ovulatory follicles and hydrated oocytes in female ovaries. This work was with local fishers who used bottom hand lines and involved FK. The citation is on the bottom right and highly suggest taking a look at this and other work that's happening in Brazil on a variety of spawning aggregations of luchanids and ceranids groupers. FYI, in the northern part of this study, FSA areas were found just outside the boundaries of a large MPA. This will be discussed a bit further in the session 4 presentation on management. We also wanted to talk briefly on connectivity among regions and other FSAs. We are fortunate to have these slides provided by Dr. Claire Paris as an example of snapper connectivity patterns developed by biophysical modeling from multi-species FSAs in Cuba. These slides summarize work from 2005 to 2019 in 4 papers. The figure on the right shows the 8 regions of Cuba that were examined in the study. Every one of those regions has identified multi-species sites with several species of snappers. The text on the left provides an overview of primary objectives of this work. Findings on the next slide. First, mapping of larval pathways from Cuban snapper FSAs. Refinement of multiple modules for the connectivity modeling system. More on that in a minute. Decade scale biophysical ocean model runs, examining factors including timing, depth effects, and other features. MPAs, FSAs, and pathways of connectivity were examined in later papers, as well as many other aspects of biophysical connectivity among these FSAs and neighboring countries. Summary findings include, across the decade scale studies of Cow et al., reference on the bottom left, retention and this was shown also in the earlier Paris et al. Papers actually, because some of this work is overlapping with other work around the Caribbean. Retention within Cuba and among the 4 Cuban regions was typically higher than export. Exported larvae from northwest and north central FSAs primarily went to the southern Bahamas. As you can see on the figure on the right, if you look for the northwest Cuba and north central Cuba columns, those are the first 2 on the left side of the figure. Purple is Bahamas, light blue is Cuba. The majority of these animals, in most cases among those 2 regions, were retained in Cuba. When there was export out of country, as you can see in those blue plots, those are years went to the southern Bahamas. The southeast coast FSAs produced larvae that primarily reached Jamaica and Haiti. Larvae from the southwest FSAs primarily reached the Cayman Islands. If you look at the figures, you can see on the right, yellow is Cayman Islands, green is Jamaica, black is the island of Hispaniola with Haiti on the left side. Wish there was more time. Refer you to these papers by Claire Paris's lab at the University of Miami. And to follow through on that, this lab has developed an open source CMS, connectivity modeling system. There's a tremendous amount of information available for bringing this kind of biophysical modeling tool to particle dispersal questions you may have. We encourage you to use the connectivity modeling system in your potential work. In summary, 77 species from 5 families in the SSGSG have an estimated total of 326 potential or confirmed FSAs. The Lugonidae and Lethronidae have the most FSAs. Most of these are from a relatively small number of genera and species. From earlier, the majority of these potential FSAs are from FK, have confidence ranks of 1 and 2, low and medium, and they await confirmation. There are many research projects available for graduate students to go further with the existing first steps and information we have to determine if these many potential FSAs are indeed producing the eggs and larvae we hope they are, and how to best protect that for future generations. Thanks to many people for this work, particularly our various co-authors across various studies. Workshop funding from Cordio and the IUCN Species Survival Commission is also highly appreciated. The assistance of the Big Fish Initiative is highly valued, and great appreciation from many to Dr. Rodolfo Collado. Thank you. Sarah and I would like to thank colleagues in many regions and at Florida Tech for assistance on this work. I also wanted to mention that Dr. Rodolfo Collado, formerly of the Institute of Oceanography in Cuba, turned 85 years old yesterday. He is doing well and sends his warm regards. Our next talk is by Dr. Will Heyman, Senior Scientist at LGL, and is on spawning aggregations in terms of environmental drivers and geomorphology, a topic he has deep experience in.
Fish spawning aggregations, environmental drivers, and geomorphology. Transient fish spawning aggregations concentrate fish from large areas. And I'm going to give you a bit of detail on this based on Rick Nemeth's wonderful 2012 paper. He used this diagram to show the spatial and temporal scales of transient aggregations. Transient spawners typically live in an area over 1,000 square kilometers, but during their spawning season, they will migrate tens to hundreds of kilometers into a staging area where they'll stay for weeks or months, and then ultimately into a courtship arena where they'll stay for days, an area typically less than 10 square kilometers, but ultimately move to their actual spawning site where they're only going to be staying and spawning for a matter of hours. Using this same document to further the explanation, mutton snapper, as an example, spawn during April and May and June, their spawning season, and they'll spawn typically around or after full moon, and typically will spawn in 5 to 7 days after full moon, and will typically spawn near sunset. Similarly, in Cuba, mutton snapper aggregate in the April, May, June time, but there are some variability both from the Virgin Islands, but also within Cuba. Here you can see 4 different regions in Cuba, and the graph will show you the spawning times by region based on actually national fisheries landing statistics from 1962 to 1985, but back-checked with Fisher's knowledge in biological studies. Interestingly, some fish use similar seasonal, lunar, and diel cues, but not tidal cues. In Belize, in the Western Caribbean, we've got an example here where the spawning season goes from around March through September. And during that time, those fish spawn monthly after full moon, but they'll spawn for 4 to 5 consecutive days. And during that time, the tides change dramatically, and yet spawning always takes place right around sunset in those 4 or 5 consecutive days. Therefore, there's no tidal signal, which is not a huge surprise given the microtidal range in that area, only 15 centimeters. Let's move to the other side of the world. Here's an example from Palau, where there's actually a 2-meter tidal amplitude, and you'll see that the drivers have some similarities and yet some differences. In this example, we're looking at 2 species that spawn at exactly the same site during the same season, February through March, but at different moon phases. Here you'll see one species has a peak spawning of over 40,000 individuals that takes place about 6 or 7 days prior to new moon, whereas the other species tops out at about 6,000 or 7,000 fish several days before full moon. Adding to this story, there are diel and spatial patterns. Again, 2 species using 2 different resting sites, but migrating daily to the same spawning site. Spawning begins around sunrise in their peak spawning time, but interestingly, each species varies their spawning time according to tide, current speed, and current direction. So in the first day of spawning for this species, timing of spawning occurs right around sunrise, a little after sunrise, but in the subsequent several days, spawning occurs 90 minutes later each day, so that by the 4th day after spawning begins, it will occur in the late afternoon. It's timed to correspond with the tidal current and tidal direction. So each day of spawning, though it occurs at different times, each day of spawning, the spawning occurs when the currents flow towards the southeast or offshore, directly offshore. The pattern is different for the other species, but we're not going to get into that at this point. In addition to temporal cues, we're also going to see spatial cues, specifically looking at geomorphology. Transient fish spawning aggregations tend to occur at abrupt discontinuities in the seafloor bathymetry. Shelf edges, reef promontories, estuary mouths, tidal passes and channels, and other areas that include seamounts or ridges and artificial reefs. No pattern explains everything. In 1996, this fisherman, Eloy Cuevas, alerted me to a site that's been fished since the 1920s, a place called Gladden Spit, and we began working there and continue to do so to the present. What we showed that you can see multiple species aggregate to spawn in relation with this reef promontory. Wondering if that was common, we looked at the sea bottom geomorphology of multi-species spawning aggregations throughout the nation of Belize and documented that most of these places do indeed occur at the shelf edge at convex reef promontories. And this work was done in collaboration with the Belize Spawning Aggregation Working Group. And as a result of this, in part, Belize protected 11 multi-species aggregation sites, but Virginia Burns will tell you more about this story in the next talk. Looking wider afield, we did an analysis. My PhD student, Shinichi Kawabara, led an analysis of 108 sites throughout the wider Caribbean, determined that 80% of those sites were described as occurring at shelf edges, many near reef promontories, some at channel mouths, and several, the geomorphology was uncategorized. Multi-species aggregations in Cuba also occur, many at shelf edge reef promontories. 2 examples here, one in the Cabo Corrientes area and the other in Corona San Carlos, again, where multi-species aggregations were documented at reef promontories at the shelf edge. This pattern led us to believe we could predict aggregations, and indeed did so one time at this lighthouse reef aggregation, where we predicted this aggregation based on geomorphology, satellite images, and the temporal patterns that we were aware of from the species that occur throughout the country. Switching gears completely, this is an example of a spawning aggregation that occurs at an estuary mouth here in Texas. It's a seasonal aggregation that occurs for 6 weeks between February and March with no lunar periodicity. But interestingly, this aggregation spurs a seasonal migration of tourists that go there specifically to catch these aggregating fish. Another excellent example, again from Palau, other side of the world, multi-species fish spawning aggregations occurring at a reef channel, indeed migrating out to the reef channel mouth to form these aggregations of 3 different grouper species concentrating their aggregations at this reef channel mouth. Summing when and where are transient fish spawning aggregations? Well, there's lots of environmental drivers, a lot of variability. No pattern can really explain everything, and there's still a lot to learn. Thanks so much.
Thanks very much, Will. There are many issues raised here that can be discussed through the rest of the workshop, including our question and answer session after the next 2 talks. As a reminder, you can enter questions in the Q&A tab, and our panelists will try to answer as time allows. It's easier for us if it's in the Q&A tab and not the chat, as Fadila mentioned earlier. The next presentation is by Virginia Burns from the Turnef Atoll Sustainability Association in Belize, and is on the outstanding story of the Belize Spawning Aggregation Working Group.
Hello, I'm Virginia Burns Perez. I work with the Turnef Atoll Sustainability Association, and I'm currently the chairwoman of the Belize Spawning Aggregation Working Group. I'm delighted to participate in this global workshop on fish spawning aggregations and to share about our national working group with you. Belize is on the northern coast of Central America and is bordered by Mexico to the north, Guatemala to the west and south, and the Caribbean Sea to the east. Belize boasts the 2nd largest barrier reef in the world and a network of protected areas, both terrestrial and marine, where 13 spawning aggregation sites are protected. But long before these 13 sites became protected, spawning aggregations in Belize could be traced back to a multi-species site at Key Glory, also known as Emily, which is along the main barrier reef south of Belize City. Fishing dynamics in Belize changed when the sturdy sailing crafts became available after 1911, with origins most likely from Cuba. By the mid-1920s, these sailing boats allowed for crews of 4 to 6 fishers to transport equipment such as materials for shelter, holding pens, drying racks, and supplies like curing salt and water to the remote site of Key Glory for extended stays around the full moon in December and January each year, when groupers would be spawning. Catches of Nassau grouper remain high throughout the 1950s, with annual catches of over 45 kilograms, and by the 60s it is documented that Nassau groupers were fished at a rate of 2 tons per day. It was the most Honduras, which was the name of Belize pre-independence. By the 1960s, spear guns were in grouper population, reef fish population. Of 4 to 6 fishers to transport equipment such as materials for shelter, holding pens, drying racks, and supplies like curing salt and water to the remote site of Key Glory for extended stays around the full moon in December and January each year, when groupers would be spawning. Catches of Nassau grouper remain high throughout the 1950s, with annual catches of over 45 kilograms, and by the 60s it is documented that Nassau groupers were fished at a rate of 2 tons per day at Emily, and this was the most caught fish in British Honduras, which was the name of Belize pre-independence. By the 1960s, spear guns were introduced in Belize, likely impacting the Nassau grouper populations as it did many large reef fish populations around the world. It is documented that more than 300 boats converged on Key Glory during the 1964-1965 fishing season. By 1975, it was estimated that approximately 1,500 Nassau groupers were aggregating at the northeast point, Glover's Atoll, so declines can be inferred as this was one of the most caught fish at the time. This recognition of local declines in Nassau groupers spurred early efforts in management, including a ban on fish traps inside spawning aggregation areas. By 1987, the first marine reserve in Belize was established at Holchan. In 1991, there was a draft national management plan specifically for Nassau groupers, and throughout the 1990s, several other marine reserves were established. Unfortunately, several of the early management efforts were either not implemented or enforced, and by the late 20th century, declines in Nassau grouper landings motivated extensive surveys across Belize, revealing that only 2 of the 9 traditional spawning sites had aggregations of Nassau groupers in the thousands. This led to the first national multi-organizational workshop in 2001, including government, NGOs, academia, and of course fishers, making an urgent call for the protection of spag sites. In 2001, we saw the first scientific study on population assessment and telemetry, which predicted that the Northeast Point aggregation would be eliminated by 2013. The national workshop led to the inception of the Belize Spawning Aggregation Working Group, and in 2002, the group advocated for the full protection of 11 known spag sites and a 4-month closure countrywide for the Nassau grouper from December 1st to March 31st. In 2004, the working group helped to develop the regional monitoring protocol that is still being used in Belize and referenced throughout the Caribbean. This was followed by a national training for working group members conducting monitoring in their respective sites, and the establishment of an online database system. In 2008, strategic spag patrols were prioritized between NGOs and the Belize Fisheries Department during spag season. In 2009, additional legislations were introduced in Belize, including harvestable minimum and maximum sizes for the Nassau groupers. Nassau groupers had to be landed whole, and all fish fillet were to be landed with a 2 by 1 inch skin patch. There was also a ban on herbivores. In 2012, 2 additional spag sites were protected. The Belize Spag Working Group continued to focus on capacity building, and in 2012, another national training was organized, and we were fortunate to have Dr. Yvonne Sadovi share her wealth of knowledge with the working group. From 2013 to 2019, members continued to monitor their respective sites and enter data into the national database. This information was used to develop education and outreach materials, such as annual newsletters and posters, and a TV spot was also created for the public, reminding them to respect the fishing regulations, in particularly the closed season. In 2016, the Wildlife Conservation Society introduced the laser sizing calipers to the working group, and most members adopted this in their monitoring. Of course, we all experienced COVID-19, and so the group was inactive from 2020 to 2021. In October 2021, the Turnef Atoll Sustainability Association convened a meeting to get the working group going after COVID. Members were still very much interested in the working group, and we were also able to get a couple fishermen back on the group. We updated our video for the public and featured one of our wonderful Belizean fishers who served on the National Fishery Advisory Board and a committed SPAG working group member for many years, Mr. Carlton Jack Young.
My name is Carlton Jack Young, and I've been fishing in these waters for over 40 years. I was a member of the Fishery Advisory Board who makes decisions on these fishing areas.
The working group continues to work hard to produce newsletters and annual reports, and in November 2023, we were able to conduct another national training for our new young field divers. We were very fortunate to have Dr. Will Heyman lead this training.
This is my first PAG training, and
I've been to a few trainings before, but I'm excited to be here and to share my knowledge that I have gained over the past 15 years and to learn new strategies and to learn from others as well.
My name is Gabriela Ogarte. I'm from Belize Audubon Society. Very excited to be here. We're looking forward to our upcoming spawning aggregation season. So it's been very, very helpful to learn all the historical work that has happened and how we move forward from here.
Hi, my name is Kevin Novello. I'm the Conservation Science Manager here at TASA, and this is my first official SPAGS training.
It was critical for us to ensure that fishers participated in the SPAG training. The fishers who sit on the working group are not scuba divers, but they have a very important perspective and experience to share. Mr. Martinez, chairman of the Hopkins Fishermen Association, was able to participate in the training. Listen to his experience.
Snappers, another fish that do spawning within certain area, where they were allowed to fish certain area. Now there are some changes in respect, especially for NASA grouper in specific area. And I keep on urging fisher folks, my peoples and even people out there, to keep and protect this species. Okay, we can do better in the future. And for our generations, which is students or kids and other future fishermen, to know what's going on within the waters of Belize. Again, discipline within the sector of fishing is very, very important. That is one of my cries when I travel through this country for fisher folks. Heed what is illegal and do the right thing. Thank you very much.
The Belize SPAG Working Group continues to meet quarterly, and members monitor SPAG sites and exchange observations and experiences. It is a group of passionate Belizeans working hard to safeguard our SPAG sites. The group is actively working on maintaining a national database. We're working on updating our logo to reflect groupers and snappers, and we are currently updating our spagbelize.org website. We continue to focus on capacity building, and we continue to integrate technology into the monitoring and management of the Belizean fisheries. A very big thank you to all those who have contributed of their funds, their time, their passion to the Belize Spawning Aggregation Working Group.
Thanks very much for that, Virginia. What an excellent example of long-term innovation from A to Z, all the way through protection with adaptive management associated. Thank you. Our last talk is by Dr. David Fairclough, Principal Research Scientist with the Department of Primary Industries and Regional Development in Western Australia. This talk is on the spawning aggregations and fishery management of the sea bream, Chrysophrys auratus, in West Australia.
Hi there, my name is David Fairclough. I'm the Principal Scientist for demersal fishes in Western Australia, based at the Department of Primary Industries and Regional Development. Today I'm going to talk about Chrysophrys auratus, a large sparrow that occurs across Australia and northern New Zealand. And I'm going to talk about the fish spawning aggregations and the management of them. So what do we know about Chrysophrys auratus? As I mentioned, it's a large sparrow and it's widely distributed in warm, temperate and subtropical waters of southern Australia and New Zealand. Its common names include silver sea bream, but in Australia it's typically referred to as pink snapper or snapper. There have been many biological studies across Australia since the 1970s. More recently, using single nucleotide polymorphism analyses, we've looked at genetic stock structure across Australia, and that work identified 5 genetic stocks across the continent. In Western Australia, there are 2 main stocks, a Midwest stock and a Southwest stock. There have been many studies of the biology, as I mentioned, and this species is quite a large and long-lived species, reaching 130 centimetres and over 40 years of age. It's a non-functional hermaphrodite, and its maturity varies spatially. Adults typically associate with reefs and adjacent soft bottoms down to about 300 metres, but typically they occur on the continental shelf in less than about 100 metres. This species does aggregate to spawn, but in Western Australia that only occurs in some parts of its distribution, which are conducive to local recruitment. The juveniles then use sheltered bays and inlets. It's a really important species in commercial and recreational fisheries across Australia, and as a result and due to varying exploitation levels, the stocks sit at varying levels across the continent. In WA, it's been fished for over 100 years, with some stocks being depleted or recovering. In terms of spawning aggregations, how do we know about them? These aggregations on the west coast of Australia in Shark Bay and Cockburn Sound have been known about for a long time. In Shark Bay, they occur in about 40 metres of water to the north of some of the main islands along the coast. In Cockburn Sound on the lower west coast, they're quite visible on the surface in this shallow protected embayment. In both areas, there's been a long history of commercial and recreational fishing, which helped us identify the importance of these areas. And for example, back in 1904, 550 tonnes of commercial snapper were landed in Perth. Fishing for snapper is also widely advertised on the internet and also through social media. So how are these aggregations being managed? As I mentioned, there's been a long-term commercial fishing of aggregations in Shark Bay. 79% of the annual catch is taken in the months with the greatest frequency of mature and spawning snapper. It's also taken in a fairly small area. Records of commercial fishers demonstrate that the vast majority of catch, or the largest catches, come from reporting blocks just to the north of some of our coastal islands in Shark Bay. Over time, the heavy fishing has led to declines in catch and catch rate, and also declines in biomass as shown on this figure. Ultimately, management has been used to bring down the total allowable catch to allow recovery of these stocks. To recover stocks, more recently as biomass levels fell below limit reference points, the department worked together with fishers, the peak bodies, the West Australian Fishing Industry Council and Regfish West, the peak body for recreational fishers, to introduce a spatio-temporal closure to commercial and recreational fishing during that spawning period, and this commenced back in 2018. In Cockburn Sound, peak spawning typically occurs at about 19 to 21 degrees Celsius, which is spring in temperate Cockburn Sound. Spawning peaks after high tide at night, and the eggs and larvae are entrained in a wind-driven gyre that occurs at that time of year in Cockburn Sound. So the eggs and larvae settle out and remain in that embayment for about 2 years before moving out to coastal reefs. Because of this widely known behaviour, recreational fishers have known about it for a long time and also became concerned about heavy fishing that was occurring in the late 1990s. At the same time, there were declines in west coast demersal species more broadly along the west coast. And so the peak recreational fishing body, Regfish West, lobbied the state government to manage these aggregations specifically. This led to an initial 6-week closure to fishing during the spawning period in Cockburn Sound in 2000. Since then, fishing has continued, with charter and recreational fishing occurring in specific areas outside Cockburn Sound, and fishing basically targeting pre and post spawning fish. A study was conducted using acoustic telemetry, and this confirmed that a single migration route was being used to and from Cockburn Sound for these large fish. That study led to work between Regfish West and the state government to agree on the extension of the closure, both spatially and temporally. It actually occurred several times over the years up until 2023. The closure now occurs from the 1st of August to the 31st of January and is shown on the map. That closure aims to cover the migration period and both the roots and aggregations themselves. A range of other work is being conducted on this species being so important in fisheries. There is work going on with respect to the proposal to build a port in Cockburn Sound, and that's looking at a range of things including stock enhancement methods and how we might monitor releases, more work on stock structure and the localised nature of that, using methods to survey the aggregations or testing them, such as acoustics and drones, and also looking into climate change and how resilient this species may be to that. Many people have been involved in both the research and management of snapper in Western Australia, and we are extremely grateful to our scientist team, recreational and commercial fishers, Regfish West and WAFIC. Thank you. Are there any questions?
Thanks for that interesting talk, Dave, on this very important spirit. A remarkable amount of collective research for decades has occurred in Western Australia on this species. Very appreciated. So folks, that ends the presentation component of session 2. Thanks to all. Now let's go to the question and answer period. We're right on schedule. It's about 30 minutes after the hour for most of us, and we've got about 30 minutes. Speakers from the sessions and other experts are now available as panelists during this following Q&A. From Fadila's guidance earlier, please use the question and answer box to submit your questions. We have a variety of folks who can also engage in conversation that might be of interest in the absence of questions, but we'd love to hear from you. We will answer as many as possible live during this session. However, due to time constraints, we may have to answer some questions via text. So we do have one question that has come in that I'm going to go ahead and read this question, and Dr. Kevin Rhodes, one of our panelists, will be providing some of the answers. For Marissa Nuttall, while this workshop is focused on spawning aggregations, I wanted to ask if the presenters had any comments or considerations they would add when looking at predictable non-spawning aggregations, such as seasonal feeding aggregations. Do the vulnerabilities of these aggregations differ significantly? What different considerations do these aggregations need for management? Thank you. Dr. Rhodes.
Thanks, Ken. Yeah, I think there are some differences. I think one of the main differences really is that the spawning aggregations tend to be persistent over longer time periods. They're predictable. They usually form in the relatively same space, whereas feeding aggregations tend to be more mobile. And probably moreover, the most important difference between the two is that during the spawning aggregations, fishing actually usually occurs when the fish are still carrying their eggs. And so when the fish is removed, not only are you removing the fish, you're also removing the eggs. And so in terms of population persistence for the fish itself and reproductive output overall for the aggregation, the impact is much larger than if you're fishing on a feeding aggregation. Of course, both have impacts to the fish populations, but I think in terms of the spawning aggregations, the impact is much greater.
And I think you're muted.
Sorry. Thank you, Kevin. Anybody else have anything to add to the panel, from the panel? Okay, then we also have a question from Dr. Kafayat Fakoya from Nigeria, now in the United States. Are there problems in developing capacity building with FSAs? My thoughts are that we may be witnessing a decline in fisheries biology as a field of study. Perhaps it may not be unconnected. It may not be unconnected with the lack of interdisciplinary studies that characterizes pure scientific research for a relatively long time. What are strategies to collaborate with the social sciences for greater impacts? That's a very interesting question we could spend a lot of time on. I'll take a start on this, and then if Mitch Lay is available, Gliding Memorial Award winner of the Caribbean Network of Fisherfolk Organizations, maybe he can add to this as well. It is very true that quote, pure sciences have been very disconnected from the social sciences, and that has impeded our ability to make progress in the most amplified ways on FSA science and management. I think this workshop and many, many other workshops and research articles and projects for a couple of decades, certainly, not long after the words of the lagoon, a lot of hard scientists began to think more about social sciences, and decades later, that has certainly steamrolled a bit. So I would suggest there's a lot more awareness of the social sciences and the anthropological realities of, forget about the letters after your name and just get out there and talk to people who know it. And as Mitch Lay was saying earlier, who work at 24/7, sometimes almost 365. So I think there's a lot of progress, but I also think that all of us would agree that we're nowhere near where we would like to be. There are still far too many barriers between communication between scientists and fishers and managers. And we all need to work harder at that. There are more programs on this in universities. There are funded projects that are still robust, but we need to do a lot more. Mitch, would you like to add anything?
Well, I can make a few comments, Ken. So I agree with you, but I also would add that perhaps earlier I asked a question about are the protocols, etc., and indicators for identifying all the stages that are involved in FSAs. And the reason for doing that was part of what this question is asking. How do we build capacity in terms of FSAs? And I asked the question particularly bearing in mind that within the fishing community, we also would love to build capacity. And we have our own way of building capacity which is directly relevant to fishers, because as fishers, we engage in the capacity development process ourselves. So we would love to have the information. Then the other area that I'd love to discuss in relation to this particular question is the issue of how do we conduct research, and I mean from inception. And one of the things that have come up in the past that we perhaps can visit is the idea of collaboratively driving the research agenda. So we get together before the research occurs and we decide on the objectives of the research. And that would, if we bring the actors, fishers, fisheries managers, scientists, academia together, then we will have an interdisciplinary sort of approach which satisfies specific objectives, sustainability, responsible stewardship, etc. So I do think that we need to focus a little bit on how we set the research agenda and then the collaboration during the research that follows to achieve specific results. So I hope that adds some thoughts to the discussion. Thanks a lot, Ken.
Sure, thank you, Mitch. Very useful. Melita, would you like to address anything in addition here?
Sure, thanks, Ken, and that was great to hear from you, Mitch. I think, I just wanted to add a little bit to Kafayette Fakoya's question about building capacity. That was one of the key objectives of this workshop. So we've been recognizing that there's a real need for it globally. We're hoping to provide more training in 2025. We'd like to do it, but it does need funding. It seems to be quite hard to get funding for this sort of training. We've got certain places in the world where we really need that training in situ. We'd like to roll out some of the manuals and toolkits at these. And we've heard earlier that the use of exchange visits is really powerful. as a training tool. We certainly found that in Eastern Africa, not on this particular topic, but on other community-based research that we do. But I think that, you know, we're keen to do that for sure. So in terms of, are there programs to develop capacity building? I'd love to know if there are, and then maybe they'll fund us to do this because we're keen to do it. But I think in terms of your suggestion that the sort of interdisciplinary approach is, I wasn't terribly sure about that, whether you mean that it's because of that there's a decline in fisheries biology. I'm not sure that I see a serious decline in fisheries biology in Eastern Africa, where I come from. What I do see, though, is not a lot of interest in actually getting in the water in Eastern Africa. And I think that that's a very stimulating part of obviously not working on deep water fish FSAs. But, you know, I think a lot of the early work that was done on them was by people who dive or snorkel, and fishers who also get in the water. So I think we do need to build that interest and capacity, if you like, in younger scientists. But yeah, we're very keen to do it. So here's a community, if you like, that wants to do this, and we'll talk about that a little bit tomorrow, I think. Thanks, Ken.
Sure. We don't have any other questions right now that I see. I was wondering if any of the panelists would like to add anything. Winston, you've got a lot of experience in the Middle East, and anything to add from that region for us, please?
Yeah, sure. So on the capacity building, a couple of years ago now, we developed the IUCN guidelines on fishers knowledge, and within them, you know, there's all these different case studies, successful case studies from all over the world of how people have done this and done this right. And I think that's one thing that's really come out of the presentations today. We saw examples from the West Atlantic to the Pacific, Caribbean, and also in the Middle East. So there's so many good examples out there, and there's so many good people who know how to do this. I think one of the key outcomes of this, you know, this workshop is a pretty solid collaboration moving forward, and perhaps we need to add that capacity building element or formalize it in some way. But certainly blown away by the experience in the room. I've learned a lot from everybody. So thank you.
Sure, thank you, Winston. I wanted to remind folks, we've still got approximately 15 minutes of Q&A here, but this is the end of the second of two pairs of sessions that occurred on Monday, day one of this event. So we had a session 1 and session 2 coupled for 3 hours early today. Then we went to later today, another session 1 and session 2, which is ending now. Just FYI, folks, tomorrow is the last day of the workshop, at least in terms of this format. The Whova site and other resources will remain live. But I wanted to point out to folks that, in case you're wondering, tomorrow there'll be a session 3 and a session 4 that will begin at 1100 hours GMT, 1100 GMT. Session 3 and 4 will begin. And then after 3 hours, there will be a break, and that session will be repeated tomorrow. Session 3 and session 4 from 1700 hours to 2000 hours, 1700 to 2000 GMT. Okay, that's straight from the workshop flyer. Anybody else have any questions or comments on the panel? We just don't have any from attendees right now.
Just in session 4, we delve into fishers knowledge and transitioning it through to policy and management measures. And we've got some really, really good case studies from all over. We go into detail in the guidelines. We look at the definitions of fishers knowledge. We look at the international law and policy framework and why you should gather that, and also governance, different formal and informal governance structures in terms of making that work. So hope to see a lot of you at session 4 tomorrow, because I think, yeah, we've got a lot to learn from everybody here.
Thank you for that, Winston. Mitchell had his hand up. Please go ahead, Mitchell.
Yeah, hi, Ken, thanks. I wanted to just throw this out to the panel, and this is about fishing. So when we look at spawning aggregations, I see that there's not a lot of research done on how or what point would you say that it's fairly responsible to fish this fish that is here. So if we have a spawning period of, like, say 5 to 7 days, then at the end of 7 days, what happens there? The fish starts to disperse back to whatever area they're from. So what sort of a analysis or what sort of recommendations are coming from the scientific community? Are we saying that we just close this area for the entire spawning period, or are we saying that maybe there may be some scope for some fishers interaction, and particularly based on stock assessments and biomass studies, etc.? Some thoughts on that I'd appreciate.
I think that that's a very interesting question, and I was going to suggest that Will might want to start the answer to that one.
I certainly can give it a stab. I think one of the things that's come out of some of the early work that Melita did with Jan Robinson and that we've done in the Gulf of Mexico was looking at the range of vulnerability of different species and different places and different fishing activities. There's no magic bullet is the first question, Mitch, and no answer that's a one size fits all answer. But there is a range of vulnerabilities by species based on their life history. And I mean, for very general generalizations, I mean, I think a lot of the groupers are more densely packed, less time. Those aggregations are tighter. More of the SSG species are spread out a little bit more. And because of their life history, having, gonochoristic instead of hermaphroditic largely. These are big generalizations, so don't, but they're typically going to be more vulnerable to fishing than some of the SSG species. So that may give you, some indication there. But I think, there is a real range of vulnerabilities and a real need to look very specifically at each site, at each species, in terms of how to go about it. So yeah, it might be migration routes that are more important, or so anyway, I'm gonna stop there. But maybe Kevin or others, have something to add for Mitch there.
Yeah, that's all very relevant and true. I would say that another point to make, Mitch, is that for example, Nassau tends to be more vulnerable, and we have in the Pacific similar species where they have a very, very short reproductive window seasonally. So like 1, 2, 3 months. And so during those times, they're extremely vulnerable, because really the peak time when most of the fish there, when the eggs are being released, is usually 1 month of those 2 or 3 month periods. And so fishing on the aggregation at that point and directly removing the eggs in the fish is something that should be discouraged, obviously. Whereas snappers, for example, that have a longer reproductive season and have not quite as established in terms of location, they're not specific to a certain piece of the reef and associated with the reef itself. I think those are a bit less vulnerable, and the science actually bears some of that out. So, and in terms of longer reproductive seasonality, if I were going to say generally, when If there was a compromise made and say you have a 5 to 7 month spawning period, I would say the first 3 months, including the peak month, are probably the months that spawning aggregation fishing should be closed off. And then the last 2 months might be, something that I would say generally might be open. But at the end of the day, I would always say that when fish are finished spawning and when they're departing the site, because they tend to use common reproductive migratory pathways moving along the reef in a certain direction, those times would be preferable in terms of fishing than when the fish are actually on the aggregation. So I hope that provides some additional information.
Much appreciated, gentlemen. And I also want to add in that when we look at measures, management measures, we also relate management measures directly to gear. So some types of gear are much more embracing than other types of gear. So perhaps we say if we have a management measure that closes an area but allows at the end of that period some activity, then maybe that activity can be limited to say line gear only and say a certain number of hooks and things like that. But what I'm suggesting to you is that as we look at this, the science and the issue of the socio-economic realities of small-scale fishers, we need to come with something that is both sustainable but also responsible for both areas. Yes? Thanks, Kevin.
May I add something to that, Ken?
Sure.
Or yeah, respond to that. Yeah, so great points as usual, Mitch. One of the things that's interesting in some of the US examples, they've, since a lot of the aggregating fish are more towards the bottom, but these places also attract pelagics and other things that are up in the column. Not typically, but there are several different aggregation sites that have been closed to bottom fishing, but still allow trolling. That gets a little mushy real quick, so I'm not a big fan because you can drop gear 150 feet down and bump your throttle in and out of gear and you're trolling presumably. But anyway, so it just brings up some complexities, but your point is well taken. I guess the other thing that I wanted to mention on this is that we can try to dial in the seasonality on this species or that species at these aggregation sites, but one of the things that's kind of blown me away on this over the past several years is, and again, I'll talk more about this tomorrow, but there's a lot of these sites, because of their geomorphology, attract lots and lots of species, and they can be there, at different times, in the snappers in the spring, and then moving more into the groupers in the winter. And so, and then what we find is some of, for example, gag grouper, the males stay on the aggregation site all year round. So if you, and they're limiting the population. So if you just close the aggregation site for a particular piece of time, the male gags, which are actually limiting the population, get crushed. And that's in my simplistic mind, if you can show that it's a multi-species aggregation from multiple taxa, typically it will be attracting fish, from a great distance, multiple taxa, and maybe a good place to close completely all year round, in terms of its benefit. But again, everything's got to be site specific and work for everybody. But anyway, there's another bit for you to think about, Mitch.
Thanks, Will.
Yeah,
And go ahead, Winston.
Thanks, Ken. No, I was just going to jump in. I really liked David's presentation on the snapper in Western Australia, and I didn't quite catch the method they used to establish the migration route. So I don't know if anyone caught that. So I guess that's a question for the room. And on Mitch's point on the different methods, so in the UAE, after doing the big project, the stock assessment, the traditional knowledge work, it was actually decided to ban the trap, but allow hand lining, and that effectively halved the catch, but allowed the fishermen to continue. So that socioeconomic piece so important. So I totally agree with you on that alternative methods. But if anyone caught that migration piece, I'm super interested in the answer as to how they did that.
Thanks. We also have a question posted that recently came in regarding that. Dr. Fairclough is not available as a panelist for this session like he was earlier. I would refer folks to the talk by Dr. Fairclough. They used a combination of technology. I don't want to name it because I don't remember which specific technologies they were using. And they also, I think, have worked a lot with recreational fishers. These aggregations of that Sparidae, they call it a snapper, it's a porgy, are very, very apparent. And I think they've worked a lot with fishers. Go ahead, Melita.
No, I didn't have my hand up anyway, but it's fine. There is a question coming on Jamaica though, and someone needs to answer it.
Yeah,
And it's
I was just about to
not
get
me.
to that. I thought you had your hand
Okay.
up. Sorry.
No
problem.
Okay.
So I think we covered everything there. We're going to get to that last question I just mentioned. Mitchell, super important comments, and something that you made that probably should be iterated over and over is this concept of fishers and scientists working earlier in the process and actually co-designing these projects. There's a common term now in the United States, citizen science. I'm not sure how much that's infiltrated the rest of the world. I think it's getting quite common. It makes too much sense not to. But citizen science is a very foundational concept for a lot of what NOAA is doing now. And Mitch, there's a lot of follow-up that we'd be happy to have with you on this. Maybe we can get something a little more explicit with the Caribbean Fisherfolk Network. We're 2 minutes to the hour. We have a question from Candice Toussaint. Pardon my bad pronunciation. Is there any research out of Jamaica on FSAs for the species mentioned in the presentations? So that's a question for all the panel. Is anyone aware of any FSAs that have been found in Jamaica for any of the snapper, seabream, grunt species? This is an opportunity real quick to introduce Sarah Brooks, who is one of our panelists. She was the co-author with me of the presentation in this session on FSAs of the specialist group. She is the person who, as I discussed, performed the huge literature search and did a lot of analysis. Sarah, in your research, I don't recall you finding or me seeing any FSAs for SSG species from Jamaica?
No, I did not find, there was none in the SURFA database that I recall, and none in my newer search.
Your search of the Google Scholar sources. Right, and as you may recall, that Google Scholar search, along with other things we found that weren't not in either database, has generated a lot of new potential FSAs for the SSG species. Again, as we pointed out, many of them following the criteria we were using are 1s or 2s, low or medium confidence, primarily based on fisher knowledge. That doesn't mean that they're not real. That means that there's smoke coming out of those bushes, and we need to hunt it down. We found too many FSAs that people didn't know about from fisher knowledge. So thank you for that question. It would be very interesting to learn more about the Jamaican side of this. Mitchell.
Yes, thanks, Ken. I just wanted to give some information. And so the research that you guys are doing on aggregations and stuff like that also has made its way into management measures in Antigua, and I'm sure in a lot of the other Caribbean islands as well. So for instance, even though we have not identified specific FSAs, we know that it exists. We have fished them, yes? So we've actually instituted management measures to offer protection, but the management measures are not, they're across months. So they're not specific to a particular site. They're specific to the species. So there's a closed season specifically for NASA groupers, red hind and coney. And this also happens in other Caribbean countries. And that is primarily in a response to the vulnerabilities of spawning aggregations. I just wanted to share that.
That's great. The temporal closures, there's the sessions tomorrow are going to be, especially session 4, be heavily focused on management, and temporal closures can be very important indeed. Kevin, you have your hand up. Thanks, Mitch.
Yeah, just very quickly, I just wanted to make mention of the SURFA database. There may be aggregations in Jamaica, but if we don't have confirmation in some way that they're actually a spawning aggregation, we don't list them. For example, if it is fisher knowledge, if they haven't been investigated further, they don't end up on the database. And so it may be that there's some information out there. It's just that they haven't yet been confirmed and made it into either the literature or been investigated with enough confidence to be in the database yet.
Thank you. We're at 2 minutes after the hour. Anything else from anyone? We could all keep going for hours and hours, of course, but folks have other things to get to, we're assuming. Want to mention again, thank you very, very much, everybody, our panelists, our presenters, the many attendees from many, many countries. I think we've all been learning quite a bit in all directions. Tomorrow is the second day, the closing day of this workshop. There'll be a pair of session 3 and the first session 3 and 4 will happen beginning at 1100 hours GMT. It will go for 3 hours. Then 3 hours later, the second pairing of session 3 and 4 will occur from 1700 to 200 hours GMT. Big focus in session 3, which Melita will moderate with a lot of input from a lot of scientists. We've got Dr. Rhodes has a presentation, Dr. Rick Nemeth, Dr. Heyman on a variety of deeper science and research issues. And then we close session 4 with think of what's going to be a very interesting management focus, including a big focus on fisher knowledge. Folks, thank you so much. This isn't over yet. Thank you so much for your time. Keep the ideas coming. Let's go save fishes to help communities have better futures.
Thanks. Nicely done, Ken. Thanks all.
Thanks, everyone.
Yeah.
Thank you.
Thanks, Ken. Thanks, everybody. Great to see you there.